Thank you, Mr. Chairman, and hello to everybody. My name is Gorjanc, and I come from the Roslin Institute. And today I would like to present you a work that I've been working for quite a while now, thinking about what the statistical models for the estimation of breeding values should be if you use genomic information, and this directly then extends, my development directly extends also bringing in the pedigree information. So if we only have genomic information, we know we can either use marker-based models or relationship-based models. And relationship-based models have a great appeal to breeders because in essence they should just be a genomic extension of the pedigree-based models that we are all very familiar with. However, there is something rotten in the state of GBLUP because we often have to do some ad hoc adjustments to genomic relationship matrix so that it behaves nicely. And when it does behave nicely, we still need to invert it, which is computationally costly. We also hear reports that genomic predictions tend to be biased. We don't really know how to combine markers and pedigrees, etc., etc., etc. And in this presentation, I would like to convince you that the culprit of most of these issues is the way the breeding value model is defined within GBLUP. I'll propose a solution. This solution will give us a proper statistical model, which we can cast in a rather generic framework that most of us are quite familiar with, and this will automatically give us the inverses and solve quite a lot of these issues. So if we look at the GBLUP model, the way it was originally proposed was that the breeding value is defined completely by the inner product of marker genotypes and the marker effects. I am using notation W for marker genotypes and M for marker effects. Now what this implicitly assumes is that, conditional on the marker effects, there is no uncertainty in the estimate of breeding value. And we know that this is not true because markers are simply not capturing all the variance. And when we are specifying these models, we need to be careful about distinguishing between the estimated value and the true breeding value. So the model for the true breeding value should be the inner product plus some residual that is going to capture this variance not being captured by markers. So if we revise our breeding value model by sticking in this residual, what we now get is that the conditional distribution of breeding values given the marker effects is a normal distribution with expectation equal to this inner product and some variance capturing this remaining variance. Now there is quite a lot of empirical support for doing this because markers simply don't capture all the genetic variance. We often see that if we do analysis with GBLUP model, that the residual phenotypic variance tends to be bigger than with pedigree-based models. And also a lot of people have done what they have done is they added residual polygenic term in the phenotypic model, and that reduces the bias of resulting breeding values. And in essence, what I'm saying here is exactly the same thing, with the only small difference that I'm saying, put this term not in the phenotypic model, but in the genetic model. And if we do this, we get now a model without the schizophrenic effects, and so we get a proper statistical model, and we can cast it in what I call a rather generic framework. And the way I look at this framework is that we need to define a model following some domain theory, in our case quantitative genetics. For me, it's very intuitive to do this graphically with graphical model notation. So by just scribbling the circles for variables on a piece of a paper, drawing some arcs between the variables according to what I think is appropriate model, and then just following this notation, we can directly get the conditional distributions out with the conditional mean and the variance. And after we have these specifications, we can just use the standard probability theory to derive the inverses, which in this case, as shown on this graph, I apologize for all the mathematical details in this presentation, but for those of you that have been already setting up the pedigree inverse will immediately recognize that this is basically the same thing. And graphically, what's basically going on is, in pedigree model, we need to update the element of the matrix for the individual, the parents, the connections between the parent and individual, and the connection between the parents. We are doing exactly the same thing now, but now we have a breeding value, and the parental values are now the marker effects. And please note that in this presentation, I'm going to be using notation Q for the inverse matrices and notation V for the variance matrices. And also note the subscript MA So these matrices are jointly both for markers and breeding values together. So if I use this rule over and over and over for all the markers and all the breeding values, I come up with this expression for the inverse. Please note, now the marker, there is a large block for marker effects. It's basically an outer product of the genotype matrix. I get the diagonal matrix for the breeding values because conditioning on the markers, the breeding values are independent. And there is a cross term which is equal to genotype matrix because markers and breeding values are connected through this genotype matrix. Now, if I continue using this rule also for the phenotypic values considered as variables and the same for the fixed effects, I come up with the mixed model, the standard mixed model equations, which in this case are now estimating the marker effects, the breeding values, and the fixed effects. But since we have a rather generic framework and it's quite intuitive to play around with these graphs, can we easily extend it with the pedigree? Yes, we can. Just follow these simple rules. And I'm going to show you with a rather simple example having the set of markers, two individuals which according to the pedigree are supposed to be independent, unrelated, and their progeny. And I assume everybody is genotyped to keep the thing simple, and we can discuss later what to do when individuals are not genotyped, or at least everybody is not genotyped. So we've seen how to do this for founders. So we have breeding values of their breeding values. There's going to be inner product plus some residual because markers are not capturing all the variants, right? Now for the progeny, we just follow the pedigree model, which is saying that the breeding value of individual is 1/2 of the breeding values of parents and the so-called Mendelian sampling term. But I have definition for the parents, which I can include in this model. So what happens is that the parent average gets basically decomposed into a genomic part and the pedigree part. And if this progeny is also genotyped, I can do the same with the Mendelian sampling term, which now becomes split basically into a genomic part and the residual of a residual, if you like. Now, there is an important note here that this, you see the W star. This W star is basically a genotype vector of an individual. So, and from this one, I need to subtract the genotypic parent average because the genotypic parent average is already in the parent average component. So once again, to summarize this definition of a breeding value, when we have both genomic and pedigree information, the breeding value is basically genomic parent average plus pedigree parent average plus genomic Mendelian sampling term plus the residual Mendelian sampling term. And we can estimate this residual Mendelian sampling term if we have phenotypic information on this individual or the phenotypic information on the progenies of this individual, because this R star 3 term will appear also in the definition of breeding values of the progeny. And if we look at the general expression for such an inverse, what we basically see the difference now is that before we had a diagonal matrix here, now we have the pedigree relationship matrix, because when I combine both markers and pedigrees, conditioning on the markers, the breeding values are assumed to be related according to the pedigree. And also please note that all of the genotype matrices are being multiplied by this pedigree inverse. And what essentially is going on, that by doing this, in the genotype matrix, there is a pedigree structure encoded indirectly in, and by multiplying by this matrix, we basically remove the pedigree structure out of the genotype matrix. So the marker effects, at least depends on how much information we have in the data, are going to be free of the pedigree influence when we do our estimation. I'll probably run out of time, so I won't show you an example how this actually process goes on, but if anyone is interested, I can show it during the discussion. Now, it's funny how these things work out. When I was preparing this talk for this presentation, I saw a paper of a colleague Zensing Leo that came out in Journal of Dairy Science, and they basically come up with the same mathematical expressions as I did. And by reading their paper, of course, in very detail, I noticed that Nicholas Gengler came up with the same expressions at the EAP meeting a while ago. What I basically did is I was more focusing what the quantitative genetic model for estimation of breeding values should be. And once I had that properly defined by conditional distributions and by help of this graphical notation, I basically just applied the standard probability theory and came up with these inverses. Now, because it's a very generic framework, it's very easy to extend it, for example, with the IBD information, and, for example, for a bit more complicated populations that have a bit more complicated structures, such as, for example, plant or tree populations. But I need to warn that we need to be able to estimate those variance components, and if we don't have any pedigree structure in our population, we won't be able to disentangle how much is captured by markers and how much is not. So this is still an open question, actually, how can we estimate those variance components efficiently. So to conclude, what I basically presented you is a generic quantitative genetic framework that I believe encompasses both pedigree models and genomic models. Please note that the genomic model that I'm presenting here is not equivalent anymore to ridge regression on phenotypes on markers, because I'm adding residual into genotype model, but we can also combine this genomic and pedigree information together, and I believe that this should solve out some of the rottenness in the state of GBLA. In addition to my co-authors, I would like to acknowledge that quite a lot of things that I have been presenting to you here now today is I've had extensive discussions with Eduardo Manfredi from INRA and Bruce Steer from AGBO in past years. Thank you very much.
Questions?
So I have a question. So
can you hear, or can you use the microphone?
Is the likelihood of your model different than a model where I put the G matrix and the A matrix? Because I see that you have the parental genotypes, the average of the parental genotypes, and in another term you have the individual genotype minus the parental area. So these things cancel out and boils into a G matrix, and then your covariance structure is the G plus the A. So the likelihood seems to be equivalent to a model where you put the G and the A and estimate the variance components.
Yeah, it's a good observation. And so if we go back to actually this expression for the inverse, this is this complicated thing, right? So this is an inverse covariance matrix for markers and the breeding values. And if I want to remove the markers from our model, so I need to basically integrate them out, what I basically come out with a variance covariance matrix only between the breeding values, which is basically the good old G a la Van Reyden plus the pedigree part. But the good thing about this is computational aspect, that I can set up this directly, while here I can't set up this matrix directly, because I would basically need to invert this. Now, if I have only few individuals genotyped, and this thing is more cheaply than this one, but we are actually getting more and more into stage where many more individuals are genotyped than we actually have markers. So this is actually going to be it's going to be more efficient. But again, it is what I think is very good and very intuitive is that we are actually trying to understand what the model is that we are fitting, and that's why I think this has great appeal.
Okay, thank you.